big data summary. And I'd like to thank Miles Parks from Cambridge, who has really been one of the leaders in IBD genetics internationally for kicking off this webinar summer series. And so kind of the motivation for this is in one part COVID and the pandemic, and how can we get together as a community in the absence and the restriction of travel restrictions. But even more importantly than that was really this concept that we're at a particularly crucial inflection point in complex genetics generally. And, you know, where do we go from here is the perfect title that Miles has provided for us. And I think we're at the point now where big data in context with genetics really has kind of an unprecedented opportunity for making major impact. And so how we as a community get together and do this is tremendous. And so we asked Miles to kind of talk about where we go and how do we scale data and scale progress. Everyone is naturally muted. We've asked this to be kind of a relatively short talk, 30 minutes, followed by questions. So everyone is muted. And the way we can do questions then is if you kind of hover over the bottom, hit the participants button, and then raise your hand. I think that'll be a more organized way to manage the question and answer session. So Miles, thanks again.
So thanks, Judy. These are really weird times, as we would all recognize. I guess one of the bonuses is that we have to spend less time standing around airports and sitting on planes. One of the downsides to that I realized earlier on today is that it doesn't leave you so much time to get your slides together. So apologies if these are a little bit higgledy-piggledy, but here we go. I'm a clinician, I'm not a scientist, so you're going to get a rather clinical perspective on some of these aspects and less of the technical things that you can get from elsewhere. Thank you for the invitation to talk. Great to see so many colleagues from NIDDK on the meeting, and I look forward to seeing a few more of these webinars as time goes on. How do I make my slides go forwards? Let me just see what I can work out to do that.
You can also hover on the lower left for arrows, or you can use your arrow keys.
Yeah, it's not working for some reason.
Not doing it?
Sorry for this. I'm having to do this off my laptop because, okay, there we go. All right. So first of all, some acknowledgements to the many people who've contributed to the work that I'm going to be talking about. Colleagues around Cambridge in many different labs, the IBD bioresource team that we have here on site, and I'll explain in a slide later about them. Colleagues in the UK Genetics Consortium that we've been running for the last 15 years or so, and of course the many people in the International Consortium and friends and colleagues, some of whom are on this webinar today. So today I'm just going to talk about some of the challenges that we face in IBD genetics, have faced historically, continue to face now relating to the heterogeneity in IBD. We'll focus some of the talk on our original goals in IBD genetics relating to pathogenesis, and then move on to the sort of more contemporary issues, I think, relating to personalized medicine and how we might handle those before moving on to a summary. So when we all began this game back in the 1990s, had somebody said, what causes IBD? You'd have probably met some sort of a Gallic shrug, I guess. But for those of us who were getting interested in genetics back in those times, we wondered whether genetics might provide us something of a route in to this question. Many of us started with linkage studies, which didn't get us terribly far beyond NOD2. But of course, GWAS came around the corner and really got everybody going in this field. The early goals to define the susceptibility, loci, and pathways underpinning the pathogenic mechanisms in IBD, perhaps giving us something of a leg up to understanding the environmental drivers as well. And then I think we've slightly retrofitted this, but identifying new targets for drug therapy. And I think it sits okay there, actually. And it's definitely been borne out by what's happened and continues to happen. So briefly, and I hardly need to do this to this audience, GWAS, of course, provides an unbiased survey of the whole human genome, genotyping hundreds of thousands of markers in large panels of cases and controls, using stringent statistics to identify SNPs which are associated, in other words, where there's a statistically significant difference in allele frequency between cases and controls, then identifying the genes that map to those same regions, and understanding the the impact of those genetic variants on gene expression, on function of the protein product in the relevant cell types. And really from the early days, and this was our earliest UK study from the Wellcome Trust Case Control Consortium, it became clear that IBD was a fertile place to be studying. We were the clear winner in the WTCCC in terms of the number of loci identified, and that has largely remained the theme as these studies have got bigger and better through our collaborative interactions. This study led by Judy and published in Nature in 2012, identifying the substantial overlap really between Crohn's disease and ulcerative colitis at a genetic level, and highlighting many new loci. I think there were about 160 at this point, and the proportion of the genetic variants explained by each of those. And then as the clock has gone forward, so GWAS has continued to deliver really. This was a study which we produced from our UK consortium, by now 60,000 cases. This was in 2017, and over 200 loci associated at genome-wide significant. You see we've had to cut off the y-axis here. There are so many loci and the strength of association is so significant. Feel then my pain in leading a study on irritable bowel syndrome recently in the UK Biobank. Here we've got 53,000 cases of IBS, and we've pulled out a magnificent 6 loci associated with IBS for a roughly equivalent panel size. So, of course, a number of pathways highlighted. You can see this in this Wordle here. I'm not going to go into any detail really on any of these. These are all well known to everybody on this call. But I think GWAS did serve a purpose in highlighting the role of cellular innate immunity, particularly in Crohn's disease, the roles of autophagy, NOD2, and so on and so forth. Really for the first time, up until this point, much of the discussion had been focused on adaptive immune pathways. And actually another wing of the innate immune pathway, epithelial barrier function, seemingly rather selectively associated with ulcerative colitis. And then this whole plethora of different genetic associations seeming to correlate with T cell function, innate immune function, and so on and so forth. So understanding these things at a high level is all very well, and it's undoubtedly done us a big service by doing that, but there are still big challenges in the system from even the current knowledge that we have. And really for us to get deeper biological insights regarding these signals requires a more granular understanding, really to pin down the causal variants at each of these loci and understand their functional impact, not least on gene expression. A small proportion of causal variants at each of these loci are coding variants. These usually have a rather large effect size, as you can see on the plot here. The causal variant, typically if one of the lead SNPs at a given signal or a given locus is a coding variant, then typically that will be the causal variant. And usually that's relatively obvious, of course, even if the actual functional effect of that variant may be far from obvious. And there is, again, within these studies, massive scope for new biological insights. So just to illustrate this, this is the signal, one of the first associations identified in Crohn's disease, originally from Stefan Schreiber's group in Germany. I know NIDK as well came rapidly after this, as did our UK studies. And then, you know, taking typically between 5 and 10 years, but this nice study then in Nature in 2014, identifying that this common coding variant associated with Crohn's disease increases the likelihood or the susceptibility of the protein product of ATG16L1 to cleavage by caspase 3 under conditions of inflammatory or metabolic stress, resulting in reduced xenophagy and disrupted pathogen clearance. So these data really flowing from the functional insights gathered from GWAS. Here's another one even more obscure in many senses. This gene was an orphan gene identified in the Jostins et al. Study, and through a series of experiments, and I know how much hard work has gone into this in Arthur Kayser's lab here in Cambridge, drilling out the role of this particular gene product, now renamed FAMIN, in immunometabolism and its role in oxidative burst and purine metabolism. So as I said, uncovering real new biology as a consequence, a downstream consequence of the GWAS studies that we've undertaken. But, you know, these are just 2 loci. We've got 250 loci now in IBD. There's a massive amount of functional analysis still to be done here. And of course, it's true to say that most of the variants or most of the loci that we've identified so far don't contain a coding variant, or at least the coding variants are not the lead signals at those loci. And we can infer, therefore, that most of the causality of IBD, genetics at least, derives from non-coding regulatory variation. For most of the loci, linkage disequilibrium is limiting and limits our ability to map these causal variants genetically. Some have, however, been so mapped, and you'll be aware of this nice study from, again, the International Consortium, led by the triumvirate of Michel Georges in Liège in Belgium, Mark Daly, well known to all of you, and Jeff Barrett as well, and Hailing Huang as the first author on this paper. And again, one of the striking findings here is just how much biology there is still to work out. Nearly half of these loci, which were tagged to a single causal variant, mapped to regions with no known functional annotation so far. So a huge amount of biology still to be unearthed here. So I just want to draw your attention to this really nice study led by James Lee, who's been a long-standing collaborator of mine here in Cambridge, published in EMBO Molecular Medicine a couple of months ago. And James has been tackling this question of how to pin down causal variants in either gene deserts or regions of the genome where things are less well characterized. And he's developed use of a massively parallel reporter assay system now in primary CD4 T cells, and this has been a real tour de force to get this working properly, to identify causal variants from their functional effects. So just to walk you through this panel here we have, in fact, one of the loci nailed down to a single causal variant by the international study I just mentioned, and demonstrating that this SNP, the single causal SNP of this locus, does indeed correlate with, or the genetic variation of this SNP does indeed correlate with transcriptional activity on this reporter assay. Here's a locus, in fact, from Angspond with 3 SNPs in the credible set. And over on this panel, you can see that just one of these 3 SNPs shows this differential expression in this reporter assay. Bring this down now to this locus, which is a Crohn's disease locus, multiple SNPs, as you can see here, in tight linkage disequilibrium, impossible to refine these genetically. Use this reporter assay. One of these SNPs jumps out, showing marked difference in transcriptional activity in this reporter assay. The others are down here in the grass at the bottom of the plot. So here we have a nice functional assay that allows you at scale to go after multiple loci. It's a really neat system. And again, just to illustrate this point, this is one of the illustrative stories that James drew out in his paper relating to the TNF-AIP3 locus associated with Crohn's, but also a bunch of other immune-mediated diseases. As we can see in the international study, multiple SNPs, all equally associated with disease susceptibility. James identified the fact that one of these risk SNPs disrupts a NF-kappa B binding site, and then using a H3K27 acetylation studies genome-wide, was able to show that this disruption of this NF-kappa B binding site, in fact, leads to a marked abrogation of this super enhancer at this particular locus, and using capture high C, demonstrating that this super enhancer interacts with the promoter region at TNF-AIP3. So I think this is a nice illustration. Obviously, this is in CD4 cells. It is one of the leading cell types implicated in IBD pathogenesis, but showing how you can really scale up to run these studies. And I'm sure we'll gain many, many functional insights from these types of analyses to help us to pinpoint the causal variants. What other approaches should we be using, or may we use in the future? You know, I remember a study which I think, or a meeting that was convened, I think, by the NIDDK in San Diego. It must have been 5 or 6 years ago now, where we were tasked with trying to think through what would be our ideal experiments to run over the next few years to help us to understand GWAS loci and the pathogenic mechanisms that might underlie them. Thinking about then this regulatory variation, how can we interrogate it in all cells in the gut at both baseline and stimulate states of stimulation? Well, there are multiple efforts afoot in this regard. You'll be aware, I'm sure, of the GTEx Consortium, the Human Cell Atlas, and so on and so forth. There are many other endeavors as well. And the technology that's being well used here now, and we in Cambridge are running studies in collaboration with Carl Anderson down at the Sanger. I know the Broad are doing these studies and many others as well. Judy, I know you've been involved in these studies too, using single-cell RNA-seq on ileal and colonic biopsies in many, many hundreds of people in both inflamed tissue and uninflamed tissue from cases and controls. To here now correlate expression of genes in each individual cell type with genotype. And ideally, in due course, factor in the microbiota to understand how that's also influencing expression. These are big studies. You know, it's taken us 5 years plus to get these going properly. But I do think these are going to pay dividends to us over the next 2 or 3 years in terms of understanding exactly what's going on and what these regulatory networks, how they operate and how they work. So just turning back to some of the current challenges, given that we already have a truck full of loci, many of which we haven't properly characterized yet, is it worth still hunting for yet more susceptibility loci? And if so, how should we do this? What technologies should we apply to this process? Well, to the is it worth it question, it's very clear there's a linear correlation between sample size and number of loci discovered. This isn't asymptoting anytime soon. And I do believe that there are still many hundreds, possibly thousands of loci that we might unearth using bigger GWAS effectively. Of course, these loci confer progressively smaller effect sizes. We have to recognize that. But I do believe these contribute to depixelation of the overall pathogenic picture of IBD susceptibility. And that by joining the dots, it can help us in large measure, even for these small effect size loci, it can help shine a light on the relevant pathogenic pathways. To some extent, this point well illustrated by the interleukin-23 receptor pathway, first identified by Rick Durr and the NIDDK consortium, and now multiple other pathway components also associated. But there are several other pathways for which this is now the case as well. And let's not forget that this process of GWAS discovery for susceptibility loci remains an important conduit for drug discovery. And to illustrate this point, this study from our UK group, our latest GWAS, identifying a clutch of integrin genes, a number of which are already targets for known therapies that we are using in the IBD clinic. So I think illustrating the point that it is still worth us going after GWAS studies for susceptibility loci at ever greater scale. And again, to underline this point, these data from GSK published in Nature Genetics a few years ago now, and this is even more so now, identifying the fact that if a particular molecule being targeted by a new drug asset is itself implicated by GWAS studies, it stands more than twice the chance of getting successfully through phase 3 trials and into clinical practice. And of course, given the costs of drug development and running drug trials, this is a massively financially implicative finding. What technology should we be using? Well, here's me getting home yesterday evening. It's a bit gloomy, I'm afraid, in my half wooden, rather old car, as you can see. This is how I imagine John Ryu traveling home in an electric car, slightly flashier than mine. I would point out that both of these vehicles, they've got a wheel on each corner, and they both get you from A to B. Further, I would point out that, that John's flashy car over here is extremely expensive. The technology that underlies it doesn't yet really work properly. And this one leaves you at risk of standing by the side of the road halfway to where you're trying to get to. I've never been let down by that like that by my wooden car over here. So you sense that I'm a bit of a fan of GWAS. I don't think we're there yet for whole genome sequencing at scale for those myriad reasons of cost and how do we actually make it work. And kind of to illustrate this point, this was a study that we ran in the UK using, in this case, low coverage whole genome sequencing on about 2,000 UC, 2,000 Crohn's cases. And despite spending a lot of money, I've got to tell you, and I know this drove the analysts crazy getting these data out, we identified one disease gene through this major amount of expenditure and work. It's an interesting gene, but there was only one of them, despite the fact that we were reasonably well powered to identify many other of the sort of lower frequency variants that might, in our models, have contributed to disease pathogenesis. Exome sequencing, I think, is a slightly different story. It's certainly much more financially tractable. It's about 1/10 of the cost of whole genome sequencing. We have better ways of interpreting the data. I've no doubt but that it will uncover new loci and definitely also help us to resolve at least some of our GWAS loci as well. And so I think exome sequencing is a here and now study and as many people on this webinar will be aware, there is already a big international study being led by Mark Daly, and indeed with data from our UK group from the international consortium. And I look forward to seeing what that tells us. So my preference for now is of a combination of GWAS and whole exome sequencing, but put the DNA in the freezer and keep close tabs on it. At some point, surely whole genome sequencing will come, and that will be our preferred modality. But really, until we can afford to do that on tens of thousands, if not hundreds of thousands of people to deliver the relevant statistical power, I don't think that it's a technology that we need necessarily to go after at this stage. Now then, lost my pointer on my laptop. There we go. So thinking now more about some of the more personalized medicine aspects, how can we identify the genetic determinants of more clinically tractable things like treatment outcomes, disease subphenotype, disease prognosis, and so on and so forth? And we're well aware, any clinicians on this webinar will be well aware that the disease course varies substantially between patients. We have some tigers and some pussycats in the clinic. And the severity of the disease phenotype often has a bigger impact on patients' lives than the diagnosis itself, actually. So here are data from Scandinavia from Tina Jess showing that about 20% of our patients from diagnosis will experience a rather severe disease course, depending on how you define that. Roughly similar proportion, a much milder disease course. Of course, treatment response also varies. This is true of any of the drug treatments that we look at in IBD, these data from the SONIC study. And what we really want to have, or ideally would be a crystal ball of some sort to help us to predict response to treatment and disease outcomes at the time that we diagnose or first diagnose our patients in the clinic, enabling us to select the optimal and personalized therapy from outset, reducing toxicity and potentially improving efficacy as well, and improving outcomes, of course, and allowing us as well to manage our patient expectations rather more accurately. When you're first diagnosed with Crohn's disease, clinicians on this webinar will well recognize the look of fear on the more insightful ones for what the years ahead might hold for them. And this nice quote, which I've often reflected back to, the success of personalized medicine depends on having accurate diagnostic tests that identify patients who can benefit from targeted therapies. Quote from Francis Collins of the NIH, of course, so that we can identify people destined to run an aggressive course and target them with our best therapies up front, and while potentially, for those who are destined to run a rather quiescent course, spare them from the risks of unnecessary immunosuppression. I'm going to come back to that in a minute. First of all, just to spend a moment contemplating pharmacogenetics. It's one of the sort of low-hanging fruits in the genetic world, I think, in terms of clinical implementation, at least. This study from a few years ago, originating primarily in Korea, but I know Dermot McGovern, who I think is on the webinar, contributing data from CEDARS and elsewhere in the US as well, and identifying NUDT15 as being substantially associated with risk of leukopenia, one of the major side effects of azathioprine and mercaptopurine use, risk of leukopenia in particularly the East Asian population, but actually also in the North American population as well. And Tariq Ahmed, who's led a lot of the pharmacogenetics work in the UK and on behalf of the international consortium, doing an exome sequencing study, I think with Mark Daly as well, identifying also this gene, in fact, a rather different genetic variant, a deletion polymorphism, also associated with risk of leukopenia in the UK population, and showing numbers needed to genotype 100 patients. And effectively, you have to genotype everybody, and this is the direction of travel, let's face it, genotype everybody coming through your clinic and apply this knowledge. You would reduce in the UK the risk of thiopurine-induced myelosuppression by about 12%. That's a big deal. You know, this isn't these are not trivial drugs, and they're not trivial side effects. Speaking of which, pancreatitis, and again, a study led by Tariq Ahmed's group, this was a GWAS study identifying this particular HLA allele as associated with risk of pancreatitis. Number needed to genotype 76 to prevent one case of pancreatitis. What about predicting response to therapy? Well, I don't think the studies have yet come through. This was a relatively early study, beset by all the small sample size type issues that beset us in the early days of our genetics work. You'll be aware of this nice study again from Tariq and his pharmacogenetics group from the PANT study, demonstrating that for individuals who carry this particular HLA allele, A105, that their risks of mounting an immune response to anti-TNF therapy are significantly higher than in individuals who don't carry this risk allele. And bear in mind, this risk allele is carried by 40% of the UK population. So this is immunogenicity, development of antibodies to infliximab, which is usually hotly followed by loss of response to this, our most powerful treatment in Crohn's disease. These are for individuals who don't carry a risk allele. These are for individuals who do carry a risk allele and who are not on immunomodulator therapy. As you can see that for them, they are almost invariably developing antibodies to this, our most powerful drug in Crohn's disease, within a year of being started on it, a lifelong condition. Similar data, but rather lesser magnitude of effect for adalimumab. So what about future directions for pharmacogenetics in IBD? We really need to get to work and put this technology to work in our clinics. But there are some steps that we need to take in order to do that. We need to get better at collecting DNA on everybody and running GWAS arrays with appropriate pharmacogenetic content on them on everybody coming through our hospitals and clinics. These data need to be analyzed in an accredited diagnostics laboratory, not in a research laboratory. This is going to be important. I would put the question to you now, something I've been reflecting on in recent months. Is it ethical even for us to do genetics research on human samples in anything other than a diagnostics laboratory now? And I think we need to give this some serious thought. Pharmacogenetics data then needs to be stored in a place where it's accessible for everybody who's doing prescribing, and of course, embed decision support software around that as well. And we need to get better at doing all of this stuff, pushing the knowledge that we have now back out into our clinics so we can prescribe more safely and more effectively for our patients. Ideally, we want to close this virtuous circle so that we capture prescribing data and outcomes data, and we feed that back in so that we help us to develop polygenic scores, polygenic risk scores for adverse outcomes and treatment response as well. Pharmacogenetics, as I say, it's the low-hanging fruit. Listen, it costs $25 now to run a GWAS array. This is the cost of doing a CRP and a CBC, right? These data are there for all time. We can justify doing this, I think, on anybody who steps foot through our hospitals. And this is certainly one of the ambitions that we're now running out here in Cambridge. Important to capture the consent of willing individuals so that we can access their healthcare data and use, of course, the rest of the GWAS array to help drive discovery science, whether that's PRSs or exactly what it is. What about disease behavior? I'm not going to dwell too much on this. These studies are well known to people. One of the surprises in some senses is that despite having, well, at the time of this study, 160 susceptibility loci for IBD, only 2 of them were associated with the disease subphenotypes that we studied at that time. This study led by Charlie Lees and Isabel Kleinan, not 2 being associated with ileal Crohn's disease and HLA with particular colonic subphenotypes in both Crohn's disease and ulcerative colitis. And this quite nice data showing that actually colonic Crohn's disease sits rather halfway between ulcerative colitis and ileal Crohn's disease. Is there a genetic contribution to prognosis? Well, this is a study which James Lee led in Cambridge. I confess this was designed on the back of a fag packet down the pub one Friday night, but it kind of worked, which was good. We reanalyzed data from the Wellcome Trust Case Control Consortium, doing a within cases analysis, comparing individuals with aggressive Crohn's disease and mild Crohn's disease to identify genetic variants associated with prognosis rather than diagnosis. We defined severity rather empirically. We were down the pub, it was getting quite late, as fizzlers, those individuals not requiring any escalation to immunomodulator therapy or surgery and with at least 4 years of follow-up, and flarers, people requiring at least 2 immunomodulators or 2 surgeries or some combination of. And ran a replication panel actually of individuals recruited in Australia and New Zealand, where clinical practice is quite similar to the UK. Choosing the tails of the distribution, I think this can be a powerful model for some of the studies that we do. And identified 4 loci at genome-wide significance associated with disease severity. And interestingly, none of these, arguably the HLA, but actually it was a different HLA allele, none of these overlap IBD susceptibility loci. So it looks like the biology of disease severity is quite different and distinct from the biology of disease susceptibility. And James then went on to work up the FOXO3 locus in a bit of detail. Why is this important? Can we use this information in the clinic? Well, no is the answer, because the studies are too small and the effect sizes are too small. So we can't make any sense of this at the moment. But listen, what we can do is work towards making these sorts of, using the principle of what James did to get smarter about developing polygenic scores for disease outcomes in the clinic. We're going to have to do this on loads and loads of people, tens of thousands of people probably, to give ourselves the relevant power to develop these sorts of instruments. But we will be able to develop them if we really set ourselves the task of doing this. There's been a lot of talk about use of polygenic scores, polygenic risk scores for predicting development of IBD out there in the general population. Even for somebody like me who has a rudimentary understanding of statistics, that's never going to happen. The target size is too small. 0.3% of the population have got Crohn's disease, for example. It's too difficult a target to hit. Even if you have a very accurate test with a very good positive predictive value, you're not going to get there. It's a much easier target to hit. The 20% of individuals in whom you're newly diagnosing IBD and who are destined to run a severe disease course. These should be statistically tractable for us to get after and to develop the genetics that allow us to identify those at most risk of running a severe disease course. It will require very large cohorts, and we will need to get our heads around the fact of getting, as Judy mentioned at the beginning of the talk, getting better at extracting data from our electronic health records or routinely collected data, or if needs be, capturing that data through sort of manual entry. But listen, I think the world has moved on beyond that, and we need to get better at tapping the resources that are already there to help drive these sorts of endeavors. I don't need to dwell too much on this. I was just going to make the point that these sort of pragmatic definitions of disease severity still seem to stand up. I think, those of us who have sat in rooms over the years with the International Consortium, I think one of the few frustrations of the International Consortium have been those conversations around how do you define severity and define outcomes. Listen, it's too tricky. It's one of those things, the closer you get to it, the more you look at it, the more murky it gets. My view is have a couple of pints, sit down with a fag packet and just jot down what seems sensible. And you're probably going to come up with something that's just as good as any amount of carefully considered consensus meetings. We should just do it. What other realms can PRS operate in? We've already mentioned that it's not going to really work in the general population to predict IBD, and there's probably no point in doing that anyway. It might be helpful faced by a group of people presenting to your gastroenterology clinic with abdominal pain to tease out those who have got IBS from those who've got IBD. You're never going to be able to diagnose IBS genetically, let me tell you that. But you might be able to identify those who are at higher risk of having IBD underlying their symptoms, and predicting treatment response as well. And of course, this would be hugely valuable. Again, it's going to require really big cohorts to allow us to get after this, and big cohorts with appropriate clinical data capture. This is one of Carl Anderson's slides playing to this idea of trying to identify and develop polygenic scores for treatment outcomes, and then sort of reverse into understanding the genetic underpinnings of that and the heterogeneity that may exist at a genetic level to underpin these rather heterogeneous responses. Again, big sample sizes are going to be required to do this, but hey, this is the ambition over the next 5 years. What do we need to drive these clinically impactful discovery efforts? Data, 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 of course. And as I've already mentioned, we should be recruiting every willing participant for DNA and to access their healthcare records to help us to drive these new discovery efforts. Pharmacogenetics is the sort of low-hanging fruit that makes it an easy sell to people, I think. But of course, there's a bunch of other data that will generate alongside that will help the discovery science. To just show you one of the efforts that we're running in the UK to help in this regard, we've set up over the last few years what we call the IBD bioresource, run now in 100 hospitals across the UK, initially set up to help drive functional genomic studies to be able to recall patients with particular genetic makeup for rebleeding and resampling. Our initial goal was 25,000 people with Crohn's disease and ulcerative colitis. We launched back in 2016. As I say, more than 100 hospitals. We're currently slightly overachieving. We're on 34,000 at the moment. We've been slightly slowed down by COVID, unfortunately. So recruitment's at a halt at the moment for this sort of study. But nonetheless, we are characterizing all of these people using GWAS and whole exome sequencing. We have access to their clinical data. We capture their data on a rather structured clinical data sheet at the point of entry. And they can all be recalled by genotype or phenotype for downstream studies. And equally important is another effort that we're running in parallel and really based around the IBD bioresource, an IBD digital research hub. What this allows us to do is to extract routinely collected healthcare data from electronic health records, from laboratory records, laboratory blood tests and stuff, radiology records, endoscopy databases, prescribing databases, and as well all the centrally held records, which in the UK are held at NHS England. I'm sure in the States it's at whichever the health insurance companies are, but where data regarding hospital admission, surgery, death, and so on is captured. And to allow us to then pull all of these data together, all linked to patient identifiers, we then link in the genetics data, the GWAS and the exome sequence data that we've got, and we put that in a trustworthy research environment for anybody to be able to access these data to analyze. You have to apply, and your application has to be approved by the Scientific Advisory Board for the NIHR bioresource, but we're aiming to make this, facilitate this process so that this large data set is available for discovery science and to test hypotheses and so on and so forth. So we're very excited about this. We're just getting going with this program at the moment. Again, it's been slightly slowed down by COVID, but we have got 10 hospitals identified who are all beginning to feed data into this resource. So in summary, and I'm sorry I've overrun a little bit, GWAS discovery is very much still live. New targets for discovery are coming into view, relating, I think, significantly to polygenic scores and development of those close to clinical outcomes. Functional genomics remains challenging, but that definitely will reveal new biology. We should align genetics research and data science, a point Judy made right at the beginning of the talk, and embed both of these in our routine clinical care. This is what will drive the large data sets that we need in order to make sense of the next steps. Molecular stratification tools are coming, and I keep telling all my clinical colleagues this. Most of them glaze over at that point, but whether they like it or not, I think we should make the point to them and start using them. You know, for clinicians who are willing to do this, we should help them to use these tools. And I think very quickly it'll become apparent that this is the way forward, and that patients that are being looked after by forward-thinking clinicians will have better outcomes and safer outcomes, and this will then sell itself at that point. So on that point, I will hand over to you, Judy, or hand back for any questions anybody's got.
Thank you so much, Miles. That was both a practical and visionary talk. So for the group, everyone is naturally muted, but hit the participants tag at the bottom, and then raise your hand, and Colleen and I will call you.
Judy, I imagine you're going to run this as a conversation, because you've got many people on the audience here who are at least as well qualified as I am, and it would be great to get a discussion going actually as to what you guys are up to and how you see the future as well.
Sure, certainly. So speaking of a great colleague, I see Rick Dorr. Let's unmute Rick. Okay, Rick.
Hi, Miles. I can say hello to you now that I'm unmuted, but that was a fantastic talk. I just wondered philosophically about one thing. I mean, you mentioned a couple of times that you think there's still room for more GWAS, provided we have sufficient sample size. You also mentioned that there's really no hope for predicting, you know, development of Crohn's disease, but maybe using genetics to predict outcomes once you know a person has the disease. I guess my question is, do you think that the loci that have been identified in case control studies are going to be different or the same as the ones that modify disease and predict outcomes in the future? I mean, do we need to do GWAS within case for all these different phenotypes?
Yeah, I would say that we do. I don't know that they necessarily will be the same. And, you know, the early studies, the one that I quoted from James Lee, suggests that these loci can be different. Now, you know, it may be that as time goes by, that these, what we've identified as prognostic loci, as it were, will be uncovered as susceptibility loci in due course. But at the moment, they look to be quite different. And I don't know that there's necessarily an expectation that the pathways of disease severity will be the same as disease susceptibility. Actually, intuitively, to me, it makes sense that they might well be different. Actually, what do you think? You're muted.
Oh yeah, we have to unmute Rick again. Oh, sorry, Rick. Maybe I can unmute him.
Colleen, can you unmute Rick? Sorry.
There we go. I think thank you. No. I think I agree with you, Miles. And you know, basically the fact that we haven't really, you know, found in some fairly large studies you know, a whole lot of association with subphenotypes, which I guess in some way is an outcome.
Yeah.
It makes me suggest that they are different, but it could also be that the granularity of our phenotyping, you know, has not been there either, and we might need to do some molecular phenotyping or something as well,
Yeah.
in addition to the clinical phenotyping. Yeah.
Yeah. I mean, we don't know for sure that disease course is as genetic as susceptibility is, I suppose, and those studies, I guess, will be tricky to do. I mean, there are some clues about that, aren't there? If you look at the, you know, colectomy rates are sometimes familial. I think some of those early data from back in the mid-90s suggested those sorts of things, but the data is less robust for the heritability of disease course than it is for susceptibility.
Yeah.
So please introduce yourself, and then, so Mark Silverberg next.
Hi, Miles. It's great to see you, and I always enjoy talks a lot. My question for you is, again, philosophical about the microbiome. So how do you alluded to it a little bit, but do you have sort of a specific thought about how microbiome data can be integrated into GWAS studies and what its role is going to be in all those areas that you talked about, whether it's disease prognosis or a therapy response? What are your thoughts on how we would do that as a individually and as a consortium?
Yeah, as you well know, many of us been around the houses lots of times on this as to how to do it. I think, to my mind, trying to work out the microbiota of disease susceptibility, I think we've been defeated by that. And certainly whenever we write a grant about it, we just get told you can't disentangle cause and effect, and so go away. So we have gone away and given up applying for those things. But we have actually, Chris Lamb, who's an excellent young investigator up in Newcastle, you may know, working with John Mansfield, has recently got a big grant from the MRC to do a study looking at microbial predictors of response to anti-TNF therapy. And I think those are the sorts of studies where we do stand a chance of making some headway. I think, you know, there is these early data from Ramnick's group looking at vedolizumab and one or two others looking at some of the other newer biologics. I think we're underpowered at the moment. Those are sort of exploratory analyses, aren't they? But again, I think we at our peril risk underestimating how important the microbiota may well be in this regard, in terms of, you know, if you look at the checkpoint inhibitor data in melanoma response and all that stuff, you know, there's some powerful things going on in the microbiota, aren't there, influencing response to medication. And so I think starting with those sorts of studies, getting the data collected around those, and then seeing how well we can integrate that. The study that Chris is leading on relating to the microbiota is going to do metabolomics as well on blood, urine, and stool, and integrate the GWAS data as well. And we'll see where we get to. Of course, this is going to be machine learning territory for sure. I was listening to a talk on machine learning a couple of weeks ago and was impressed by, I think it was a study in rheumatoid arthritis that was looking at transcriptional signatures and where the presupposition had related to the disease phenotype and the drugs would correlate with gene expression. And actually it was alcohol consumption that correlated with gene expression, nothing to do with disease. And this kind of, you know, really, it told me we've all got to be really careful about how we interpret these data. It was a real lesson, actually. It was a great talk about how, you know, if you're going to put in lots of data, get as much data as you can and don't overlook things. Include all the data that you can in these sorts of studies. Just, it will help you to tease out what's noise and what's real signal. How about you, Mark? What are your endeavors in the microbiota?
Well, I think the analytic challenges are daunting, but I also think back to how I remember speaking about IBD genetics in 1995 or whatever it was, and people saying those, you know, you're never going to overcome the statistical challenges for untangling a linkage analysis or GWAS approach. And you know, look where we are. So I think we need, I agree with you just on the point that it's critical when you see what's happening with IBD in Asia and South America. You know, there's a critical environmental component, and I think we're obligated to figure out a way to integrate it into our approaches. And it's a challenge, but I think we need to do it. Yeah,
I'm impressed by detailed longitudinal studies on even just small numbers of people. And hopefully the GEM study may shine a light on some of these pathways, I guess.
So again, questions from trainees encouraged. Next is Dermot McGovern.
Dermot hasn't been a trainee for 25 years, Judy.
We're just setting you up, Miles.
Dermot and I trained together, I know that.
Can we unmute Dermot, Colleen, if he's not? Yeah.
Great. I was unmuting, but I kept being muted again, maybe so that you could get that joke in. Yeah, Miles and I did train together. Miles, I really enjoyed that. Thank you. I've been trying to get my colleagues in the States to have lab meetings down the pub and write things on the back of a fag packet. And just to translate that for people, that's a cigarette packet for years now. I just wanted to go back to the pathology and think about more objective markers of disease severity. So I think in that hub that you were talking about, are you also planning to get access to pathology and then even more than the pathology reports, actually images of the pathology where you can start to do some of the machine learning approaches to pathology, the same with radiology. So we can think about perhaps part of our problem with the clinical phenotyping is that one, we're relying on humans to do that. And the second is that perhaps that's a little too late in the day. We're too near the sort of, and there are too many things going on towards the end stage of some of the clinical phenotypes that our patients have. And we need to think about other ways of capturing that data.
Yeah, it's a good point, Dermot, and thanks for raising that. I agree with your sentiments entirely there. So the IBD Hub, yes, indeed, will capture digital radiology data, not just the reports, although it will also capture the reports because, of course, that for machine learning purposes is quite helpful, having all of these reports read by a consultant to run against those things as well. So that's a powerful resource in itself. Digital pathology, I don't know what it's like in the US and North America, but most of our pathology in the UK, I'm afraid, is not yet digitized. There are a couple of forward-thinking hospitals where that is happening, and we are absolutely capturing that data. We're trying to work out how we can fund that separately to get digitization of at least the diagnostic slides when the patient first presented. And I think that would be, as you say, a powerful driver for machine learning potentially. How are you set in the States? Do you have digitized pathology at Cedars or not?
Yeah, we do, and we've sort of invested in this significantly from an IBD perspective, and we're beginning to find some very interesting things, and obviously you can start to automate that, and then you can get high throughput and so on. And so we've been really interested in this area since we started working with Thad Steppenbach and looking at panel cell morphology. Yeah. And tying that into both environment and genetic and clinical outcomes. So I actually think it's a really interesting area, and I think that it's something that we as a community need to think about if we're going to solve some of the problems that you've highlighted.
I agree.
So Miles, let me ask two questions while we wait for additional questions from trainees. So the first question is, let me push back a little bit on how important it is to identify causal alleles, because, you know, one of the limitations of MPRA as opposed to direct ex vivo analyses is that you may find the causal allele for that activated T cell in that condition, but it's possible that it may be causal in innate cells in a different condition. So that's kind of the first question. The second question is, given our next week's speaker, Jonathan Pritchard, who's kind of founded this omnigenic principle of most of the genome contributes in most polygenic traits. How do we think about, okay, you have genetic support, and how do you kind of go from the genetics to drug discovery if most of the genome contributes?
Yeah, it's an interesting, I mean, I think there is an empiric approach to this, isn't there, whereby you only have to look at the proportion of drugs that there are currently in clinical use which do target GWAS signals to know that we're doing something right. And so the sort of philosophy of there's so much of the genome contributing that actually you might as well target anything. Well, that can't be, that intuitively doesn't feel right from a sort of clinical perspective. this idea of how important is it to define the causal variant, I think is an interesting one, you know, and in the way that when you're preparing a talk, you do sometimes think more carefully about these things than perhaps otherwise you do. I don't know that it is so important to, you know, if you're going to use the sorts of single cell sequencing and correlating that with genotype, is it really important that you correlate it with the specific causal variant, or actually anything in LD with that will do effectively, and it will help you to get to the answer. So I sense that you're right. I think there might, we run the risk of it just being too complicated to properly get our heads around and too difficult, and we shouldn't obsess about it too long. If we can get to the answers without having to worry about that, then we should stop worrying about it to some extent, I think.
Thank you. Any other questions from the group?
So we have one other person, Saad Shaikh. I'm going to unmute you and you can ask your question.
Oh, okay. Oh, hi. This is Shazad, University of North Carolina, Chapel Hill. This was a really great talk, but I wanted sort of your insight on two things. One, how do you think, I guess, single cell functional genomics will play a role in the context of where we're headed with genetic association? And I think the second is looking for, I guess, treatment links or responses with variants in other diseases that have been deeply sort of studied using GWAS, and how that sort of can overlap with IBD GWAS, like say RA or psoriasis.
So I guess it's an interesting question, isn't it, as to what extent the determinants of treatment response may be overlapping between Crohn's disease and ulcerative colitis, and indeed between, you know, IBD and, as you say, rheumatoid and psoriasis. Are we expecting the same factors to determine response across those different disease phenotypes? I rather am, I have to say. For the same reason, I would slightly expect that some of these determinants of disease severity, IBD severity, may be the same determinants of severity in rheumatoid or psoriasis. And they may be the inverse of things correlating with, you know, clearance of infection and so on and so forth. And this is kind of how the world works, isn't it? And there are some data around that. Now I know James Lee was involved in developing some of that stuff around his FOXO3 workup, where he showed that that SNP also correlated with adverse outcomes in rheumatoid arthritis, but also correlated with reduced clearance, I think, of malarial parasites, I recall from his paper. So I do think that these are telling us more generic truths outside of IBD as we begin to focus on those things. With regards to the single cell and correlating that with genetics, is that going to definitely get us somewhere? I don't know. But it has to be worth a go. And, you know, again, I go backwards and forwards on slightly to Judy's point a minute ago about is it really important that we nail the causal variant? Is it really important that we nail the cell type in which Crohn's disease originates? I think it's probably naive to suppose that it's only one cell type, would be my guess. But, you know, in the way that you know, so many of these conditions seem to be stemming back to the stromal cell, or the thing that nobody ever thought of. You know, I think the beauty in many senses, I think, of single cell is, of course, that you capture data on all of those cells which nobody ever thought of, and it may shine a surprising light in a way that that nobody had ever really anticipated. So I don't know for a fact that it's going to be useful, but it's definitely worth looking, because you know, if we're blind to it, we run the risk of walking over the top of something important.
Thank you.
Thanks for the question.
Okay, so last question goes back to Rick. If you give a second question, or did you not go down? Yeah, it must be back to you, Rick. Can we unmute Rick Dorr? Okay, go ahead, Rick.
Oh, okay. Am I unmuted now? No, I actually didn't have a new question, just to follow on to the last conversation. That, you know, I think it's really appealing to think of this potentially as a situation where there might actually be actually some organ you know, specific susceptibility factors that determine the initial early susceptibility to the disease in whatever organ it is. But then once you've got this dysregulated adaptive immune response, you know, that can be perpetuated because of the dysregulation, it might be that a lot of those genetic factors are shared across many different immune-mediated diseases. And that's the way I think it might actually, you know, there might be some truth to that, right? So.
Can I ask, to what extent in your hospitals in the States are you able to, are you thinking about doing GWAS at scale and just recruiting everybody, putting everybody into GWAS studies and just having it at the front door of the hospital pretty much? You doing that, Judy or Rick?
We're thinking about it.
Yeah.
I just had a meeting with pathology about like making sure the DNA just goes through the CLIA hands first, and then you so you maintain it and can return results. So yes, we are moving forward on that.
Yeah, yeah. And these, you know, these have the potential. Then I think if it's just part of what happens in hospital, this is how we'll drive the sorts of sizes of data sets that we're going to need to really get a handle on some of these things. And that would be very exciting, I think, if we can. There are clearly some arguments to be won, and people need to be won over to the fact these data can be held securely and that they, you know, that they don't represent a threat to them. But I think if we can win that argument and make the case, then these will be very powerful data sets for discovery science.
Okay, I think that's a wonderful last note, Miles. Again, I'd like to thank you for a really wonderful talk. And next week, Jonathan Pritchard. Bye, everyone.
Thanks, everybody.
Thanks, Miles.
Thanks, Darren.